everybody? I think we can get started. Certainly feel free to shuffle around, grab some food if you haven't, but let's get us started. So I'm Kathy Adella. I am a physics professor at Mount Holyoke College. I am the host for the evening, although you won't hear too much from me other than the acknowledgments and some announcements. We're here to hear Scott Garman tell us about curing the common cold and seeing things that we have no business seeing in some ways, certainly with the naked eye. So let me get a sense first of how many of you have never been here before? That's fantastic. An extra special welcome to you, but an extra special welcome to our regulars for coming back time after time. SciTech Cafe is a science cafe, and the idea behind the science cafes are that we bring a scientist, that would be Scott today, into an informal setting, that would be Amherst Brewing Company, to talk about science with the general public. So you do not need a science background, and one of the special things about this event compared to going and watching documentaries in museums is that you can actually ask questions and interact and get answers to those questions that you have been always wondering about. So we encourage questions, ask lots of questions. If you've asked 3 questions already, maybe go and see if your neighbor wants to ask your next question so that we don't get too many questions from exactly the same people. But this should be an interactive process. And there are demonstrations today, and Scott has been here many times. He's a regular with his family, so he knows what people are expecting and what he wants to do. How does the Science Cafe, SciTech Cafe, come here? Well, we have funding largely from the National Science Foundation, through the Materials Research Center on Polymers at the University of Massachusetts, through the Center for Hierarchical Manufacturing and Nano Center at the University of Massachusetts, as well as from community-based learning at Mount Holyoke, which is bringing academic learning, classroom learning into the community. We have our last event of the year. We take a break for July and August, but it is going to be on Monday, June 9th. This is breaking the regular pattern, so Monday, June 9th, because we're trying to avoid Memorial Day. And this is going to be UMass physicists fold under pressure. And they're going to talk about origami. And they're doing some really fascinating things with origami, understanding both the theory behind origami and using it in the types of materials that are actually biocompatible, and what is it that we can actually do with these materials. So that's going to be Thomas Hull, Chris Santangelo, and Ryan Hayward, who will be here in about a month. 2 other events next week. There is another science cafe in this area, the Organismic and Evolutionary Biology Cafe. Their next event is Sex and Drugs and Planets and Bugs. That's going to be Monday, May 5th, and at Essalon Cafe if you want to go attend there. And then I am actually doing a Science on Screen at Amherst Cinema next Tuesday. And that is the movie Fantastic Voyage from 1966, which is a story about miniaturizing a crew and a submarine to go into the body to then use a laser, which were relatively new, to cut out a blood clot, and then the adventures that follow. It won awards in 1966. It's delightfully dated now in a way that when I rewatched it, right, it's not early CGI, it's not early computer effects, it's like they built things out of felt and cardboard, not cardboard, but they actually had to like create this inner world. And the introduction, the first 20 minutes, is going to be about, well, what can nanotechnology do today that actually targets medical interventions inside the body? Can we send mini robots in the body to do things? So that is what I have, and with that, I will pass it off to Scott Garman.
Okay, so today we're going to talk about a spectrum of things from a spectrum of sizes and a different spaces we will talk about. So everybody should have received a few pipe cleaners if my kids have done their jobs correctly. I want you to make a nice long linear polymer of your pipe cleaner. So while I'm talking, feel free to weave yourself a nice long, it turns out our pipe cleaners are going to represent a polypeptide, and a longer polypeptide folds up into more interesting shapes than the default 12-inch pipe cleaners. It turns out it's very hard to find 3-meter pipe cleaners. So let's get a sense of the scale we're going to talk about today. Leonardo da Vinci drew this famous picture of Vitruvian Man. And a man is on the order of, this man is on the order of 2 meters. Okay, so 2 meters is is about this big. And what we're going to do today is get down to what we call nanometer scales. So we have to increase our magnification by, say, 10 orders of magnitude. So the first thing we're going to do is take a little trip in orders of magnitude. So if we look in the middle of our man and zoom in on that, our man has lungs. Lungs are on the order of 0.2 meters, something like that. We are going to zoom in to the bronchi. Bronchi in diameter are a couple of centimeters. Okay, that's 2 orders of magnitude we've gone down. So now we're going to go down into the bronchi, into the tissues in the lungs, and we get to the alveoli. Alveoli are on the order of a couple of millimeters, something like that. Little air sacs in the lungs, and now we've done 3 orders of magnitude. Okay, zooming in on those air sacs in the lungs, we get here are the air sacs in the lungs. Air sacs in the lungs. We can start to see cells when we've done 4 orders of magnitude. We're at 200 microns, or 0.2 millimeters, of magnification here, and we're starting to see cells. So let's zoom in on those cells. And when we're at 20 microns of magnification, the 5th order of magnitude, we start to see individual cells. Okay, those individual cells have features and so forth. We're halfway there. So here is what looks like a sphere, but no, in fact, it's a cell. We need to look inside the cell when we're interested in what's going on inside a cell. So what we're trying to do is use the techniques of physics that we've borrowed to look at a biological problem like what's going on in a cell.
Do you mind saying, are those true colors?
Those are not true colors. So we are actually at a resolution where color is no longer, at some point, once you get past the, say, 700 nanometers of visible light, colors actually drop off. So this is actually a false color image. It's actually from an electron micrograph, but this artist, Ann Weston, has colored it in a very beautiful way. Okay, let's zoom in a little bit more. If we look inside a cell, we get, so here's a piece of our cell right here. And these dark little guys with the V's, these are viruses. These are influenza viruses that are binding on the surface of a lung epithelial cell. And we're now at our 6th layer, our 6th order of magnitude in magnification. And we're going to take a look at those viruses. At 200 nanometers, or 0.2 microns of magnification, we start to see individual viruses. It turns out those viruses have a bunch of nucleic acid material on the inside and a bunch of proteins on the outside. And these spiky proteins are doing the job of the virus to help get into the lung epithelial cell. One more expansion. We're going to look at a cross-section of our virus. So now here's our nucleic acid. And again, this is not an electron micrograph. This is an artist's depiction. But inside the flu virus, we have nucleic acid material that propagates the genetic information of the virus. And on the surface, we have these spiky proteins. The blue one is called the hemagglutinin. The red one is called the neuraminidase. They have crazy names, but it turns out they do functions on the surface of the cell. The hemagglutinin, its job is to bind the lung epithelial cells and get the virus particle inside the lung epithelial cell. The neuraminadese's job is to act as an enzyme to cut off some of those receptors on the surface of the lung. Okay, let's look at the neuraminidase, and we're going to zoom in So now we're down to 2 nanometers of magnification, and we're looking at an individual protein. It turns out the neuraminidase from the flu virus is a tetramer. There's 4 symmetric objects arranged in a regular 4-fold symmetric pattern. And again, the job of this protein is it acts as an enzyme. It cleaves off receptors that are on the surface of the lung epithelial cell, and that prevents a flu virus from reinfecting the same cell that it just came out of. One more level of magnification. We zoom in one last time, and now we're at the level of atoms. And at the level of atoms, we can do chemistry. So here is, this is called the binding site, or the active site of flu neuraminidase. And it's got all these features in it. So these are now the electron class, the surface of the flu virus that's determined by the electrons that are associated with the atoms on this protein. So this compound that has the sticks those are exploratory compounds that are being soaked into this enzyme in order to treat the flu. So this is experiments where the investigator is testing compounds to inhibit the flu virus from infecting and propagating in lung epithelial cells. Okay, so I think that was 10 orders of magnitude of resolution. So what we've done here is now we're at the level of individual bond distances. So a carbon-carbon bond is, say, 0.15 nanometers. And what we're going to do today is take that distance and we're going to expand it from here out to Grace's and Caroline's cousin's house in California. We're going to take a 2 centimeter distance, stretch it across the country to California. That's the level of magnification we're going to be talking about today. Okay? How are we going to do that? How are we going to get that level of magnification? Well, we're going to go visit Bizarro World. Okay? Who knows anything about Bizarro World? Any fans of Superman comics from the 1960s? What's Bizarro World? An alternate universe. Okay, in Bizarro World, the Earth is not spherical, it's a cube. In Bizarro World, there's Superman, but there's also this character called Bizarro Superman. And Bizarro Superman, you can see the S on Superman is frontwards. The S on Bizarro Superman is backwards. And if Superman is good, Bizarro Superman is not good or evil. In Bizarro World, there's a Bizarro code where we do the opposite of what happens on Earth. So we spend most of our time in non-Bizarro space, but today we're going to visit Bizarro space. What else happens in Bizarro World? It turns out Batman. This is Batman. This is what is his name? His name is Batzarro, I think. So he is the alter ego of Batman who lives in Bizarro World. And you can see his bat is upside down. He's got some bad teeth. And it turns out his utility belt has chewed up gum and gum wrappers and trash in it. I learned quite a bit about Bizarro World. Seinfeld visited Bizarro World. I don't know if anybody remembers the classic Seinfeld episode, but here is real world Jerry and George and Kramer, and here are the Bizarro World equivalents of them. And these characters are nice characters because they live in the opposite universe of the Seinfeld characters. So here's some things that I put together, the difference between real world and Bizarro World. So that cubic-looking Earth is actually Earth spelled backwards. What is small in the real world is big in Bizarro World, and vice versa. Things that are right side up in this world are upside down in Bizarro World. There are these characters in the real world who have their alter egos in Bizarro World. And then we have something in this world, the time domain. In Bizarro World, we're going to think about the frequency domain. And specifically for crystallography, we live in real space, but we call it reciprocal space, right? Reciprocal is upside down. Okay, so we're ready for our first experiment into Bizarro World. I need a volunteer who wants to visit Bizarro World. You won't get hurt. There should be no lasting impact, but I haven't tested this experiment fully. Gracie, would you like to be this is my daughter Grace. She signed the release. Okay, I would like you to stand here. You're the visitor to Bizarro World. I need somebody who is a good detective. Do we have any people that are good detectives who aren't going to drop this very expensive lens that I borrowed from Mount Holyoke College? Do we have any very responsible volunteers? Come on up. All right, Gracie, I'm going to light you up on your head here because we need light to visit Bizarro World. No, not too close, please. Don't look at it. Elsa, can you hold this with 2 hands and not let go? All right, here's my investigator into Bizarro World. I need one more person who's our detector in Bizarro World. Want to be our detector? Yeah, come on over. So Elsa, watch out for these cords here. But I need you to get relatively close to Grace. And I need you to hold that up. And right like that. And I need you to hold this paper out here and see if we can see an image of Grace. That is quite a lens. That's quite a lens. Hold it there. Oh, there it is. Hold it still. Okay, we got the lens. And in Bizarro World, it's over here. Okay, so I need you to hold this and take that paper over and see. Wait. So look at the paper. Sarah, maybe you can help. We're trying to image the light. That's it. There it is. Okay, so what do you see over there? Maybe back it up a little bit. And is it right side up or upside down?
Upside down.
Yeah, it's upside down. So when we visit from real space, okay, so I think that would work for our demo. Let's take a break and let's explain what happened. Let's thank our volunteers. Okay, so when you shine some light at an object in real space, that light bounces off the object in real space and enters Bizarro World, or as scientists like to call it, reciprocal space. So we have this space that is the opposite of real space that then we can use a tool like a magnifying lens to capture that scattered information. It captures the light that's scattered, that's off, right? So the light that's scattered off of Grace is invisible. We capture that light with our convex lens, and over here we make an image of what we scattered, right? So this lens is a tool that takes us from reciprocal space, that scattered light that's invisible, and it makes an image back in real space that we can see. Okay? And again, as a souvenir of our trip to Bizarro World, the image gets inverted. Okay? There's also a way of going to Bizarro World. Here's a photograph of people in my lab from last summer. And of course, at Six Flags, there's a Bizarro Superman with the backwards S. And here are some scientists in my lab who are doing experiments about visiting reciprocal space or Bizarro World. So we do research every day in our lab, just like this. So we can actually make a schematic diagram of this experiment we just did. So here we have some light. We shine a light on an object. That light comes off the object, and our lens is our bridge between real space and reciprocal space, or real world and Bizarro World, if you will. Okay? And then we get this upside down image. So we're going to use these principles from physics to image stuff, except the object we're looking at is not the size of a human. It's going to be this 10 orders of magnitude smaller object that it turns out we're going to use these same principles. Okay?